All right, we'll get started. Yeah, welcome back everyone to the second oral session for the day. For those of you who I haven't had a chance to meet yet, my name is Rabia. I am a PhD candidate in the Hines Lab at McMaster University, and I'm going to be the moderator for this session. So just a reminder before we start to please use the Q&A tab that's on the 4Waves window for any questions that you have for the presenter. And attendees can also upload any questions they have using the arrow that's like right beside the question that they would like to upload. And you can also continue to use the chat function for other forms of communications on the side. So with that out of the way, it is my pleasure now to introduce our first speaker, Phil Lauman. He did his BSc in the Integrated Science program at McMaster University and is now completing his PhD in microbiology and biotechnology with Dr. Jonathan Dennis at the University of Alberta. He tells me that he is an avid hiker and has just completed something called the 3 Crown of Waterton Challenge, where he almost got hit by lightning. So welcome, Phil. We're certainly glad to see that you were able to make it and you are good to go.
Okay, thank you, Rabia. And thank you everyone for coming to my talk today. So today I'll be talking a little bit about one of the organisms that's really responsible for a lot of the antimicrobial resistance, and it's contributing a lot to the antimicrobial resistance crisis, which is, of course, why many of us are in this field. And so those are the bacteria that make up the Burkholderia Symposia complex, or BCC. This complex is comprised of about 25 species, which are very related to each other, and they're part of the gram-negative genus Burkholderia. And so these species have a lot of beneficial roles in the environment. For example, they're responsible for being involved in plant growth promotion, nutrient cycling, but they're also implicated in a lot of plant and animal diseases, including human diseases. So for humans, especially humans that are immunocompromised or those with cystic fibrosis or chronic granulomatous disease, these infections are actually quite nasty, and they have a high mortality rate as a result of something called Symposia syndrome. And so these bacteria also happen to have a high rate of both innate and acquired antimicrobial resistance, which is shown in this diagram here. And so that makes them kind of an ideal target for phage therapy. Now, when we think about phage therapy, we tend to think about these phages that follow this canonical sort of lytic cycle, right? So they just make, as we all know, they make copies of themselves and then they burst out of the cell, killing it, and look for a new host that they can infect. But although the field sort of focuses largely on these lytic phages, a huge proportion of the phages that are out there in nature actually follow this alternative lysogenic cycle as well, where, as we know, they're able to integrate their DNA into the host and then they replicate passively with it before conditions change, causing them to reinduce into the lytic cycle. So today we'll be talking about these phages, which I term lysogenization-capable phages. So why do I call them lysogenization-capable? Well, so in the Dennis lab, we hypothesized that although there's a global sort of determinant of lysogeny, which is actually having the lysogenization cassette, the set of genes that permit this behavior, the tendency to form lysogens under particular environmental conditions depend on a whole repertoire of what we call local determinants of lysogenization. So these can be environmental factors, for example, such as temperature, nutrient availability, whether or not there's radioactivity in the environment, but also phage host match properties. For example, whether there's a genetic match between the phage and the host that it's trying to lysogenize. What is the multiplicity of infection within the specific environment where the interaction is taking place? Does the bacterium have any defense systems that can protect against lysogenization? And so there's probably also other factors that also play a role that we haven't fully characterized yet. And so what this really means is that these lysogenization-capable phages actually fall on a spectrum in terms of their frequency of forming lysogens, whereas phages that are obligately lytic would fall on the extreme of this spectrum over here. I hope you can see where I'm pointing with my mouse. where they're not actually able to make lysogens at all, so they just fall at that extreme. And so if this hypothesis is true, we posit that the terms temperate and lysogenic are actually quite misleading, because they imply that the phage always engages in the lysogenic cycle, which we know is not necessarily true, and that the phage is not lytic or virulent, which we also know is not really accurate. Now, in order to actually determine whether or not this hypothesis is correct, we defined lysogenization frequency of some arbitrary phage X under specific conditions of MOI, the media type, the temperature, the particular host, as the proportion of survivors of attack by phage X that are also lysogens of phage X after some kind of prolonged period of time T, which in our case was about 48 hours. And so with that, this spectrum that I gave you can actually be mathematically quantified on a scale from 1 to 0, where 1 includes phages that always or almost always form lysogens, while 0 would be either phages that, although they're able to form lysogens, they don't actually do it under those specific circumstances, or they're not actually able to do it at all. Now, to quantify this, we set up various infections with phage and bacteria of different types under various conditions, either in vitro and in vivo. We collected survivors, and then we PCR screened them for the presence of lysogens. And so when we did this with this phage KS14, we used different forms of media. So this one here is artificial sputum medium, hemolymph of waxworms, minimal media with glucose. Then we did different temperatures, different hosts, all of which this phage is able to infect. And so what we see here is that there's quite a staggering amount of variety in terms of this lysogenization frequency that this one phage has. And so this really validates our hypothesis that, it's not a black and white situation of lytic versus lysogenic. There's a lot of different environmental factors that come into play into determining what actually happens in this situation. And so then we wanted to know, if we hold all these environmental factors standard and we just focus on different phages with different hosts, what kind of variety are we going to get? And so in this diagram over here, we see the phages that we use on the x-axis and all the different hosts on the y-axis. And so what's really interesting about these results here is that all of these phages are actually genetically capable of forming lysogens, but very few of them actually do so to any appreciable degree. And so at this point, you might be wondering, okay, but this is really just sort of a matter of nomenclature. Like, why do we really care about this from a therapeutic perspective? Well, so the reason we care about this, because as many of you know, the current paradigm of phage therapy is that only obligately lytic phages should be used, because the rationale is that temperate phages always form lysogens, therefore they don't destroy bacteria, and therefore they could transfer AMR or virulence factors. But so we know, based on the data that I just presented, that this is not really true, and therefore these two parts here are not really true either. So there's really no reason why we couldn't be using these phages in situations where other phages are not available. And so, of course, this was not really be an issue if obligately lytic phages were abundant in the environment. But the fact is that for many gram-negative species, including many that we work with, which are shown here, obligately lytic phages seem to be the minority of all known phages, while lysogenization-capable phages seem to be quite abundant. So there's no need to really limit the scope of what we can do with phage therapy if this rationale is flawed. And so what we really wanted to know is, can these lysogenization-capable phages be therapeutically useful? And is that utility really related to the specific lysogenization frequency of those phages? And so the first thing that we did is we actually quantified the in vitro antibacterial activity of these phages. So we set up simple infections in vitro, and based on the data, we actually created growth curves. And so we analyzed these growth curves using a new metric called the growth reduction coefficient, or GRC. And I'm going to explain how that works using the example of this potovirus DC1 on this particular strain of Burkholderia Synergistic. So here we have an optical density versus time curve. So in the blue, we see the bacteria alone, so no phage has been added. And then the pink curve is actually showing a situation where DC1 is involved and at an MOI of 1. So it's exactly a 1-to-1 ratio between phage and a host. So what you do here is you take the definite integral of the curve, or the area under the curve, as some people call it. And so then you compare that with the area under the curve for the control. And now you have this GRC value for this particular phage X, in this case DC1, at a particular MOI M, in this case 1. And so now you can conduct the same experiment under many different MOI conditions. And what you can do is generate a curve of MOI versus GRC. And this helps determine at what MOI this phage is actually optimized. And so perhaps unsurprisingly, many of our BCC phages, however, interestingly, not all, were optimized at higher MOIs. And so that's actually the sort of approach that we took. So we wanted to see what kind of effect these phages have at the highest MOI that we have. And so when you take a look at this graph here, so again, we have phages that we used on the x-axis, hosts on the y-axis. And so you can see that although all of these phages are capable of lysogeny, many of them have quite high antibacterial effects on the host that we tested them on. And so this tells us that these LC phages are actually therapeutically useful, or they can be. And so what we notice is that, of course, I'm not expecting you to memorize this heat map or the one that I showed before, but you notice that many of these blue tiles coincide with a lot of the yellow tiles that we saw previously. And so we wanted to see if there was a correlation between GRC and frequency of lysogenization. And so, of course, we found that there is, and it's a quite strong and significant relationship between these two variables. So here we have lysogenization frequency, and this is the growth reduction coefficient, and there's a strong inverse relationship between these two variables. And so what that tells us is that this lysogenization frequency can be used to predict the antibacterial efficacy of phages, at least for now in vitro. And that low lysogenization frequency phages, such as these ones, can be used therapeutically. It also tells us that these high lysogenization frequency phages remain pretty much suboptimal for monophage therapy. So they can't really be used on their own. And so the next question that we had is, can we develop some kind of strategy for phage therapy that will render even these otherwise useless phages therapeutically useful? So in our lab, we're working on a number of different strategies for doing that. So one strategy that I'll be talking about today in the last few minutes is the strategy of using two phages simultaneously. Now, to do this, we use this very similar approach to the one described earlier. So these were all in vitro experiments that I'm describing here. And so we constructed 1-to-1 ratio cocktails of every single possible combination of phages that we had. And then we conducted these growth experiments again. And so in this three-dimensional graph that we see here on the right, the X and Y axis indicate the lysogenization frequencies of the two phages in the combination, while the color axis is actually telling us the growth reduction coefficient of that combination. And so as you saw previously on the previous slide, some of the phages are not very useful when they're used on their own. But however, when you combine them together with another phage, suddenly they have strong antibacterial properties. And so what we wanted to know is, is the effect of the individual phages just stacking, or is there nothing going on? Is it just one phage dominating the interaction? Or perhaps is there some kind of synergistic effect that's going on? And so we decided to investigate this mathematically. Now, in the interest of saving the time that I believe I am starting to lose, I don't think I have very much left. So I'm not going to focus extensively on the math behind these interactions. However, we did investigate a number of different situations where there was an autonomous effect. So in these situations, one of the phages was completely dominating the interaction, and the other phage's effect was insignificant in comparison. And in other situations, although there was no interaction between the two phages, the individual effects of the phages did stack. And so they were able to produce greater killing when they were combined together. Now, these two sorts of situations were made up about the bulk of all the situations that we saw. However, we did see one instance of an antagonistic interaction where the effect of the two phages together was actually less than what you would mathematically expect if they were additive. And we encountered 6 situations where there was a synergistic interaction, where there was a statistically significant difference between what we saw with the combination relative to what you would predict to see mathematically if the two were additive. Now, these results are pretty exciting, I would say. So this is the first instance of mathematically demonstrated synergistic interactions between two phages. And so although we are not fully clear about how this mechanism is actually operating, what we did notice is that in many of these situations, one of the phages has a high lysogenization frequency, while the other is quite low. And so we're suspecting that perhaps lysogenization actually has some sort of indispensable effect towards the interaction that we're seeing. And so we have some data that actually supports this, but we can't say it definitively at this moment. So some take-home thoughts, not all lysogenization-capable phages are created equal. I think if you remember anything, this is the take-home message here. And the therapeutic utility of these phages has to be determined through rigorous evaluation with these mathematical tools rather than just based on genetic characteristics. So we don't really want to be painting all of these phages with the same brush just based on the content of their genome. And then we found that low lysogenization capable phages can be just as effective as obligate lytic phages, and even the high F-list phages that are not useful on their own, they interact synergistically with a lot of other phages to produce these really powerful effects. And so taken together, this shows that perhaps these lysogenization capable phages do have some role in phage therapy after all. So with that, I'd like to thank my supervisor, Dr. Dennis, and of course, all the members of the Dennis and Rabia labs for their continued support. I'd like to thank Dr. Alex Hines and all the organizers of Phage Canada for this great opportunity, and of course, all the funding agencies. And at this point, I would also like to thank you guys for attending, and I'd be very happy to take any questions. Thank you.
All right, thank you, Phil.